Discovery and development of triptans. Triptans is a word commonly used for a class of anti-migraine drugs that are selective 5-hydroxytryptamine/serotonin B/1D5H-tone B/1D agonists. Migraine is a complex disease which affects about 15% of the population and can be highly disabling. Triptans have advantages over ergotamine and dihydroergotamine, such as selective pharmacology, well-established safety record, and evidence-based prescribing instructions. Triptans are therefore often preferred treatment in migraine. History. Search for a new anti-migraine drug started at Glaxo in 1972. Studies in the 1960s showed that vasoconstriction from 5-HT-ergotamine and noradrenaline could reduce migraine attacks. Research also showed that platelet 5-HT level is reduced during migraine. Because there are too many side effects for 5-HT to be used as a drug, scientists started research on the receptors of 5-HT in order to discover and develop a more specific agonist for 5-HT receptors. Research on the 5-HT receptors and their effect led to discovery of several types and subtypes of 5-HT. A24167 showed a vasodilation effect instead of vasoconstriction due to the agonist effect on another type of 5-HT receptors, later assigned the name 5H7. A25086 was the second compound developed and showed a vasoconstriction effect but was not released as a drug due to low per-oral bioavailability. Continued research led to the discovery of the first triptan drug, sumatriptan, that had both vasoconstriction effect as well as better oral bioavailability. Sumatriptan was first launched in the Netherlands in 1991 and became available in the United States during 1993. Mechanism. Triptans are specific and selective agonists for the 5H-tone receptors. Sumatriptan binds to 5H-tone D receptors. Zolmitriptan, rizatriptan, naratriptan, almatriptan, and framatriptan binds to 5H-tone B/1D, and elatriptan binds to 5H-tone B/1D/1F receptors. Triptans are believed to exert their effects through vasoconstriction, leading to reduced carotid arterial circulation without affecting cerebral blood flow, peripheral neuronal inhibition, or inhibition of transmission through second-order neurons of the trigeminocervical complex. Receptors. 5-HT receptors are all G-protein-coupled receptors GPCR except for 5-HT3, which is a ligand-gated ion channel. The receptors that have been found to be involved in migraine are 5H-tone B, 5H-tone D, and 5H-tone F receptors. 5H-tone B are found in meningeal arteries. Agonism of 5H-tone B causes vasoconstriction in cranial nerves. The 5H-tone D receptors are located primarily in the trigeminal nerve. They are also found in vascular smooth muscles, mediating contraction. Agonism of 5H-tone D receptors subdues the release of inflammatory-inducing nerve stimulants. The amino acids contributing to the binding of ligands to the receptor are aspartic acid ASP, phenylalanine Phe serine SIR, threonine THR, tryptophan TRP, and tyrosine TYR. It has been shown that both 5H-tone B and 5H-tone D receptors in humans have a very similar amino acid structures, which demonstrates the similarities in binding properties. Design. All triptans have an indole structure identical to the neurotransmitter 5-HT. Classic triptan structure contains side chain on the indole ring and a basic nitrogen in a similar distance from the indole structure. The main structural difference of the triptans is the position of the sulfonamide and the side chain attached to it, see Figure 1 and Table 1. Rizatriptan and zolmitriptan have, instead of a sulfonamide, a triazole and 2-oxazolidone respectively. Another exception to the classic structure is seen on elatriptan, where the nitrogen alkyl chain connected to the indole ring is replaced with a dimethylpyridine, and in naratriptan, where the nitrogen alkyl chain is replaced with a 1-methylpyridine ring. One of the fravatriptan side chains forms an additional ring with the indole, resulting in a carbazole ring system. Structures of the triptans. The 5-H-tone-B/D pharmacophore, 5-H-tone-B and 5-H-tone-D receptors are considered very similar. They share amino acid homology, and their ligands expose similar binding properties, thus they have similar pharmacophore. The pharmacophore model for these receptors' ligands is qualitative and defines the relative positions of important groups. It is defined with following 5 main features: an aromatic group, usually the indole, protonated amine donor of hydrogen bond, acceptor of hydrogen bond, additional hydrogen bond site both donor and acceptor and hydrophobic region located between both hydrogen bond sites, see Figure 2. The main binding points were concluded to be the protonated amine and the hydrogen bond site. It was observed that the double bond region in the indole was necessary for the agonism in this series of compounds. Figure 3 shows how different drugs fit the pharmacophore within C and in linked analogues of 5-H-tone-D agonist. The marked sites on the figure are responsible for the affinity. The pharmacophore can be characterized as amphipathic. That means that the structure has both hydrophobic and hydrophilic groups. Relevant structural features of triptans and binding to the receptor. Triptan structures were designed from the structure of 5-HT to attain affinity to 5-HT receptors. Receptors, hence the identical indole structure. The hydroxyl group OH on the hexane of the indole core and the alkyl amine side chain on position C3 on 5-HT have been replaced with other compounds, such as sulfonamides or azole ring-structured derivatives and different amine-alkyl side chains. An electronegative group can form a hydrogen bond with THR in the pocket of the receptor. Sulfonamide derivatives attached to the hexane ring of the indole structure have electronegative properties, as well as the triazole and 2-oxazolidone on rizatriptan and zolmitriptan respectively. This can increase binding ability of the compound and the efficacy, especially with the 5-H-tone-D receptor. A schematic drawing of the binding of sumatriptan to 5-H-tone-D receptor can be seen in Figure 4. One study showed that sumatriptan fits better in the binding site of the receptor when the side chain with the protonated nitrogen atom is folded back over the indole structure. This alignment contributes to the hydrogen bonding between the nitrogen in the sulfonamine and the serone 138 in the binding site. It is also favorable to the formation of the hydrogen bond between the oxygen of the sulfonamine and THR 202. Other binding in the pocket of the binding site occurs with the nitrogen atom in the pentene ring of the indole structure of the triptan and the amino acid SIR 352. This energetically favorable position of the agonist makes it possible for additional binding of the ligand to other SIR in the binding site, along with additional anchoring between Phe in the pocket of the binding site and the indole of the agonist. The binding of Phe and the triptan is caused by p-stacking interactions of the indole and amino acid, and an additional effect on this interaction is because of dispersive effect of amino acid leucine U, not shown in Figure 4. The amino acids TRP343 and TU346 both have electron-rich p systems in their aromatic structures. With their position in the binding site, they create a sort of aromatic cage around the protonated nitrogen atom of the side chain on position C3 of the triptans. This nitrogen atom is protonated at physiological, and thereby stabilizes the ion bond the nitrogen atom has formed with a carboxylate on aspartic acid. Side chains of the surrounding amino acids can have an effect on the binding of the nitrogen atom. Mainly 3-Phe can affect the methyl groups bound to the nitrogen atom not shown in Figure 4. Elatriptan has higher affinity for the receptor, which is probably a result of the bulky substituents of the structure. The amine is protonated at physiological pH condition, triggering better uptake. The uptake rate of the agonist is different depending on whether the amine in R2 is primary, secondary, or tertiary, but the latter seem to give the best results. For the Rhone substituent, an electron-rich sulfonamide groups and amide group has shown the best results in receptor binding and activity. It has been observed that a relationship is between absorption and molecular size, hence larger hydrophilic molecules tended to have poor absorption. A small Rhone substituent is necessary to maintain the rapid oral bioavailability of triptans. By placing an electron withdrawing group or large group on position C2 on the indole structure, the 5-HT agonist is converted into an antagonist. This is thought to be because the indole ring is unable to occupy the aromatic part of the binding site. Triptan drugs. Properties of formulations. Sumatriptan was the pioneer drug in this class. The second-generation triptans, such as zolmitriptan, naratriptan, rizatriptan, almatriptan, elatriptan, and fravatriptan soon became available. Different triptans are available in different formulations and in different strengths. See Table 2. They have been formulated as subcutaneous injections, oral tablets, orally disintegrating tablets, nasal spray, and as rectal suppositories. Delivery system of the triptans may play an important role in the onset of action. The selection of anti-migraine drug for patients depends on their symptoms. The first selective 5-HT1B/1D agonist, sumatriptan, was first synthesized as a subcutaneous injection, then as an oral tablets, and more recently as a nasal spray. It is also available in some countries as suppositories. The subcutaneous injection is the fastest way to stop a rapidly progressing migraine attack. The sumatriptan nasal spray provides faster onset of action than the tablets, but it produces a similar headache response at 2 hours. Some patients prefer the nasal spray as it works more rapidly than the tablets and does not have as many adverse effects as the subcutaneous injection. Nasal sprays, although not suitable for all patients, because some patients experience bad taste and lack of consistency of response. Zolmitriptan was developed with the strategy to create a more lipophilic compound with faster absorption and better ability to cross the blood-brain barrier than sumatriptan. It is available as tablets, orally disintegrating tablets, and as nasal spray in some countries. Rizatriptan is available as tablets and orally disintegrating tablets. Naratriptan, almatriptan, elatriptan, and fravatriptan are only available in tablets for now. A specific enzyme not yet reported. The U.S. Food and Drug Administration, FDA, approved a new drug April 15, 2008, which is a combination of sumatriptan 85 mg and naproxen 500 mg NSAID. Triptans and NSAIDs work on distinct mechanisms involved in migraine and therefore may offer improved treatment when administered together. Pharmacokinetics. Pharmacokinetic properties, see Table 3, are important when new drugs are developed. Patients seek rapid onset of action to relieve the headache. Relatively short Tmax, good bioavailability, and lipophilicity are pharmacokinetic properties that have been associated with rapid onset of action. It has been speculated that good ability to cross the blood-brain barrier and relatively long terminal elimination half-life may result in a lower incidence of headache recurrence. Sumatriptan and rizatriptan undergo first-pass hepatic metabolism and result in lower bioavailability. Tone/2 equals elimination half-life. Tmax equals time to reach peak plasma drug concentration. CLR equals renal clearance. Log D pH 7.4 equals measure of lipophilicity at pH 7.4. Increasing number indicate greater solubility. VD equals volume of distribution. M equals male, F equals female. Future research. Most triptans were developed and introduced in the 1990s. Further studies have not shown much promise regarding the development of new triptans with better duration of action, efficacy, and safety profile. Therefore, it is unlikely that further variations will be developed, and new anti-migraine drugs are likely to have another mechanism of action. References